Hello friends, welcome back to my YouTube channel Chembasis. So in this video I am going to talk about AstraZeneca vaccine. So AstraZeneca vaccine is basically developed by University of Oxford, and they have partnered with a British Swedish company that is named as AstraZeneca. So the chemical name of this AstraZeneca vaccine is this one, and it is also known as AZD1222. So in this video I am going to describe how this AstraZeneca vaccine works in our body. So let's start the video. In India, this UK based vaccine has been produced by Serum Institute of India, and it is known as Covidshield. So friends, before going into the detail of how AstraZeneca vaccine work, let us understand the basic working principle of vaccine. Our immune system protects us from different types of pathogens entering into our body. However, if our immune system cannot able to recognize any new virus, then it can be fatal for our body. So basically the main aim of vaccine is to make our immune system introduce with these new pathogens. Generally, vaccine carries inactivated form of the real virus or some particular portions like protein of that virus. Some vaccine also carries some particular instructions which our body read and make some particular part of that virus. So now our immune system gets some time to have an idea that how the virus actually looks like and how to act against it. So when the real pathogens attack our body, then our immune cells are prepared to fight against it and to destroy it so that our body get protected. So this is the basic principle of how a vaccine works. Friends, earlier I have given a detailed lecture about how a vaccine works. If you are interested to know that, you can click on the above link so that you can have a very good idea of how a vaccine works. So all those things are very clearly demonstrated in that video. So let's now start with how the AstraZeneca vaccine actually works in our body. So friends, in this picture you are seeing an animated picture of COVID-19 virus. So you can see that at the central part of that COVID-19 virus there is spike protein gene. So this portion basically contains the genetic instruction of that COVID-19 virus, and when this spike protein gene enters into our body, it will instruct our body to prepare some particular proteins. And you can see that in the surface part of that virus there is some structure, and these structures are called spike protein of the virus. So this is the basic structure of COVID-19 virus. Now the Oxford based AstraZeneca vaccine is basically based on the virus genetical instruction for building the spike protein. So basically these types of Oxford AstraZeneca vaccine carries some particular instructions, and when this instruction is being injected into our body, then our body actually read those instruction, and according to that instruction our body synthesize some of the protein part of that COVID-19 virus. So this is the basic principle of how Oxford based AstraZeneca vaccine works. Now these instruction can be encoded through two forms. One is single-stranded RNA forms and another is double-stranded DNA form. Now there are two other vaccines, that is USA based vaccine, that is called Pfizer BioNTech and Moderna vaccine. So these two vaccines also carry some instructions to prepare some particular spike protein of that COVID-19 virus. However, these two vaccine basically carries those instruction in form of that double-stranded DNA. On the other hand, Oxford based AstraZeneca vaccine carries those instruction in the form of single-stranded RNA. So that is the basic difference between Oxford AstraZeneca vaccine and USA based vaccines like Pfizer BioNTech and Moderna. So friends, as I have told you that Oxford based AstraZeneca vaccine basically carries some instruction to form the particular protein of that COVID-19 virus, and that instruction is in the form of double-stranded DNA. Now this instruction basically requires some kinds of medium by which it could be inserted into our body. Now the Oxford AstraZeneca vaccine basically use chimpanzee adenovirus, which act as a vector for the vaccine which inserted into our body, and then the DNA released into our body, and that DNA codes has been readed, and according to that code some particular protein of that virus being synthesized into our body. Now this adenovirus is basically one type of virus which causes different types of fever or coughing, sneezing, those types of things in our body. But instead of human based adenovirus, they are using chimpanzee based adenovirus. So you may have some doubts or question that why they have used this chimpanzee adenovirus instead of human based adenovirus. Now friends, to answer that question, I would like to tell that the human based adenovirus is quite familiar to our immune system. So our immune system already knew about this human based adenovirus. So whenever this human based adenovirus inserted into our body, then our immune system will react very quickly and will destroy that virus instantaneously, so that virus wouldn't get sufficient time to release that genetic material that is inside this. So the main purpose of that vaccine will not be fulfilled. So that's why chimpanzee based adenovirus is being used as a vector for the vaccine, because this type of adenovirus is new to our body, so our immune system couldn't recognize that adenovirus. So that's why our this adenovirus will get some time, and within this time it will release the DNA codes of the SARS-CoV-2 virus. Now there is one thing that is important to be mentioned, that DNA is not as fragile as RNA. So that's why the Oxford AstraZeneca based vaccine do not require very low temperature to store those vaccines. On the other hand, Oxford based Pfizer or BioNTech virus basically use RNA based code, and RNA is much more fragile as compared to DNA. So that's why to protect those vaccines it require very low temperature. So that is the basic difference between UK based Oxford AstraZeneca vaccine and US based vaccines like Pfizer and Moderna. One thing I would like to actually add in this regard, that the chimpanzee adenovirus that is being used in Oxford AstraZeneca vaccine is basically an inactivated form of that adenovirus, so that whenever this adenovirus goes inside our body through that vaccine, it cannot able to increase its number further. So it is not at all harmful for our body. Now friends, let's see what happens when the vaccine is inserted inside our body. So as you can see that this vaccine is in the form of an inactivated adenovirus, which is containing that genetical instruction inside it, that is double helical DNA. Now this adenovirus basically attached to the human receptor of that human cell. So this is the human cell, and this adenovirus basically attached with the human receptor through its spike protein, and then it come inside that human cell. Now you can see that inside the human cell there is nucleus. So what happened, that this adenovirus basically released that viral DNA or the DNA that is containing the information to produce some particular protein of that SARS-CoV-2 virus. So that DNA is being inserted into that human cell nucleus. Now this DNA is being transcribed into mRNA by some process. So now it is converted into the mRNA, and now this mRNA is being goes to the membrane or the surface part of that nucleus, and then it came out of that cell nucleus and goes into the cytoplasm of the human cell. Now as you can see that this mRNA goes near the ribosome. So this ribosome is basically a human machinery. So the main basic work of that ribosome is to basically decode the different types of instruction coming through mRNA, and according to that instruction these ribosomes basically prepare different types of protein. So as I have told you that this mRNA basically consists of some kinds of instruction to synthesize some particular spike protein of that COVID-19 virus. So our ribosome basically read that instruction, and according to that instruction they will prepare the spike protein of that COVID-19 virus. And these types of basically spike protein combine to form one big spike protein. So as you can see, now what happened that these spike proteins sometimes breaks into some small fragments. As you can see, some of the spike protein basically breaks into some small fragments. Now you can see that the human cell contain both the big spike protein as well as this fragmented spike protein, and these spike protein goes into the surface of that human cell. Now you can see that at the surface of that human cell it consists both the spike protein of the COVID-19 virus as well as spike protein in form of fragmentation. So this particular pattern should be identified or recognized by our immune system. So now let's come to the next part. Now when these types of foreign particles, that is the spike protein of that COVID-19 virus, has been synthesized or formed inside that human cell nucleus, then this human cell basically send out some kinds of warning signals that they have been attacked by some kinds of unknown spike protein. So whenever these types of signal goes to the immune system, they quickly react to that signal and they will release some kinds of toxic material so that the whole human cell getting destroyed. So when the vaccinated cell dies, so it contains some of the debris of the dead cell. So these debris are nothing but the spike protein of the COVID-19 virus. So these spike protein and protein fragments can be taken up by one kinds of immune cell that is called antigen presenting cell. So you can see that these debris are basically goes inside this antigen presenting cell, and they basically introduce or present this debris into another type of immune cell that is called helper T cell. So this helper T cell can raise the alarm and help marshal other immune cells to fight the infection. Now there is another type of immune cell that is present inside our body that is called the B cell. So now when this B cell getting closer to that vaccinated cell, which contain both the spike protein as well as fragmented part of the protein near its surface. So this B cell, when came close to that vaccinated cell, some of its spike proteins are being locked by the B cell. Now what happened that the helper T cell will activate the B cell. So whenever this B cell is being activated by this helper T cell, it will start to produce antibodies. So now these antibodies, as you can see, after activated by this helper T cell, these B cells will start secreting numerous number of these types of antibodies, and these antibodies will then target the spike protein. Now these secreted antibodies can destroy the vaccinated cell. Moreover, whenever these types of antibodies are secreted, it will try to find out the spike protein that is present inside our body. Now if it will find these types of COVID-19 virus that is containing these types of spike protein, it will target those spike protein and instantaneously destroy the COVID-19 virus, so that these virus will not further attack any human cells. So friends, this is the mechanism by which AstraZeneca vaccine basically work inside our body. Now this AstraZeneca vaccine is basically given in 2 doses, and the time gap between the 2 successive doses is recommended as 84 days. And research shows that if these 2 doses are taken at a gap of 84 days, it will give the best result, and the antibodies that is being produced after the second dose is much high if the gap in between the 2 days is remain around 84 days. So that's it from this lecture, and I hope that you have enjoyed this video, and if you do like this video, please subscribe to my YouTube channel Chembiasis, and also you can like and share this video with your friend so that they could also get some information about this. And thank you very much for watching this video till the end, and I will see you soon with the next video. And till then, take care of yourselves. Bye bye.